DNA synthesis for the project is our topic. It is very important even though it's microscopic. To build our model, we decided to use candy. The colors and shapes will probably come in handy. We hope that these pics will help you remember the process, all the main functions we hope to address. Replication is important, especially for DNA, because cells cannot divide in just any old way. Mitosis and meiosis are important for the cell. If that's going to happen, DNA has to replicate as well. Once the DNA is replicated, then cells can divide. May seem confusing, but clarification will provide.
Through DNA synthesis, the cell makes an exact copy, but it has to be just right. The sequence can't be sloppy. The DNA is then passed to daughter cells in mitosis, or in the case of reproduction, to haploid cells in meiosis. DNA synthesis is crucial, as some researchers now know. Mistakes in the sequence may even cause tumors to grow. There are several studies that focus on the template strand. They think that mutations and tumor growth go hand in hand. The study says that when nucleotides pair together, but the match isn't indigenous, it has an effect on oncogenesis.
DNA is made up of a string of nucleotides, made up of A's, C's, T's, and G's that are linked to each side. At first, the DNA is just hanging in the cell. The functions are being carried out, and all is well. The steps of synthesis is a pretty long list, but the first step is for the double helix to untwist. Topoisomerase comes in and joins the strand. It helps flatten the helix and get rid of the pressure on hand. Helicase is next, and as it takes its trip, it travels between the strands, helping each side to unzip.
This separates the strands, giving them some space. Later on, new DNA can come and take its place. Single-strand binding proteins connect to one of the two secs. They make the strand irregular and help it not to reconnect. Starting the new DNA strand is a different case. There has to be a primer which is attached to primase. Once the primer is in place and the process can run free, nucleotides build off, but built only 5 to 3. The Okazaki fragments are the pieces in between. They separate the primers until DNA polymerase I intervenes. The leading strand is the one that's on the 5 end of the string, while the lagging strand is at the end, towards the number 3.
Polymerase I comes in and replaces the primers with nucleotides. Now the strands are all DNA on each of the sides. Ligase has the final job with mitosis on the brink. It goes through all the strands and all the pieces it links.